I want somebody to share with us something that you've been enlightened about today, something you were surprised about, something that was fascinating to you or you had no idea was even out there. Does anybody have something that they could share? Something new, surprising. What do you guys think?
I was just fascinated by how well mangroves are doing here. So that was a real eye-opener for me.
I know, it's exciting that something grows well. We actually had a neat discussion over in the mangrove section too today about that, about some of those views. So we need to bring that up again. All right, are we all ready? Ken, you're good to go? All right. So we have for you now the very exciting and always entertaining Dr. Ken Dunton, who is a professor here at UTMSI. He has just amazing research, both here along the Texas coast as well as in the Arctic. So without further ado, and you're welcome for that picture. I chose it myself in the bios. I took it, actually. In any case, here's Ken Dunton.
Thank you, Dana. Okay. So, just to let you know, it gets worse from here. So the title of my talk, and I'm completely to blame for this because I didn't check back on it and made a classic mistake. Blue carbon projects in Texas, coastal ecosystem processes, and critical roles in the global cycling of carbon. That's probably two courses right there. And that graphic, would you like to go through it? I just did it the other day in my graduate course, and then from here we just keep talking about geochemistry and the cycling of carbon and nitrogen, and talk about all the pathways of nitrogen cycling which are linked to the pathways of sulfur cycling, which is then linked to carbon cycling. Right? Yes. See, the grad students all know this. And luckily I'm following on the footsteps of David, Stephanie, and Steve, and I don't have to cover all this. So the real title of the talk, unless you really want to hear about all this, I'll be very happy to go through it. And I still will need to acknowledge my friends, Bo, who's been with me forever, Bo Hardigree right here, and especially Julie, who took over. When I came to the lab here in 1986, I had to take over at the kind of last minute. I took over a lab because Warren Pulich was leaving for greener pastures at the time, and I took over Warren's lab, and I didn't know anything. I came from studying kelp beds in Alaska, kelp beds on the west coast, east coast. I didn't know the first thing about seagrasses, and Julie taught me everything. So she was a very good teacher and still is, and retired and still teaching. So admin, we changed the title of the talk. Sorry, my friends at NER, but sorry, Jace, but I had to. So Victoria Congdon, where are you, Victoria? And you already met Kim. There's Victoria. She's lying low in the back. So Victoria is a grad student with us, and she really had a lot to. She put this together, and she will not give the talk. She's giving another talk tomorrow and another one next week at the meeting in Portland. So a lot of us are frantically trying to get ready for that meeting. But I thank you, Victoria, for putting this together. This project was funded by the Coastal Wind Bays and Estuaries Program, and it's a continuation of the project. Really is about is focused on the Texas Seagrass Monitoring Program. But that Texas Seagrass Monitoring Program, which I'll give a brief introduction, has produced enormous amounts of very, very interesting data illustrating patterns of seagrass distribution on the Texas coast. That program is funded by a number of agencies we'll talk about later. But the part that got added on last year, thanks to the Nature Conservancy, was blue carbon. And I didn't know what blue carbon was either 2 years ago. Now I am blue carbon. Thank you. We have our rep here from CBBB. Thank you, Ray, for supporting. So we're continuing this blue carbon. It's actually turned out to be very, very interesting, as you'll see in a few minutes. So we're going to focus. So I'm going to talk about. So by this point, you're after with Kim. Has given you a background why seagrass are important, but of course the standard answers are habitat, photosynthesis, and they produce huge amounts of organic carbon, which are food for upper trophic level animals, consumers. But a lot of that carbon, as we well know, ends up in the sediments and is stored in the plants. One of the most important aspects of carbon cycling in seagrass beds is one that's often overlooked, and that is that the oxygen doesn't just escape the plants and go to the atmosphere. A lot of it is transported through the the parenchyma, through the leaf, from the leaf tips to the roots is an open system of canals that transport oxygen or through diffusion. And that keeps the rhizosphere, the roots and rhizomes here, in an aerobic environment. And that's really important because if you don't keep this rhizosphere in an aerobic environment, what happens is that through the processes I showed in that very first slide, sulfate reduction, nitrate reduction, ammonification, and particularly sulfate oxidation, or sulfate reduction, sulfides accumulate through the decomposition of organic matter through the sulfur cycle. That produces ammonium and a lot of the nutrients, but also produces sulfides, which are toxic. When sulfides build up in the rhizosphere, in the sediments, they kill the root tissues. They're toxic. And when you drive across the Kennedy Causeway, if you live here in Corpus, during the summertime, that rotten egg smell is our sulfides being produced under anoxic conditions in those seagrass beds. Under those conditions, root biomass, rhizome biomass decreases substantially. And that is important because there's a major amount of carbon stored down here in these roots and rhizomes. I think Kim got a feel for that earlier when Kim was showing you, especially in Thalassia testudinum, the root biomass is substantial, and I'll give you some examples here in a minute just how substantial it is. So that is a very important process, and under low light conditions, which are produced, often result from dredging, ship traffic, eutrophication, algal blooms, what have you. Once you drop below 18% surface irradiance, at least here on the Texas coast, this is where our seagrasses sit. They need about 1/5 of the sunshine that hits the surface of the earth. They need it. They need to have that. Otherwise, they go into a net carbon deficit. So, so we've learned a lot about seagrasses over the last couple decades here in Texas, and worldwide we know that there's a tremendous loss. We've lost about 18% worldwide. This is from Michelle Waycott's data, and I don't have the data for the 2000s, but the decline continues. Obviously, the concerns on degradation of lost seagrasses is not only habitat and what have you, but it's release of CO2 to the atmosphere through everything we've talked about this morning, obviously, and some of those numbers are pretty big. Now, 300 teragrams carbon may not mean a whole lot to you, but later on I'll put it in perspective how big that number really is. It's for at least for Texas. So we'll come back to that 300 later, but 300 teragrams of carbon that's 10 to the 12 grams of carbon is enormous. So in the Texas Seagrass Monitoring Program, just a little brief introduction, because without this, I need to present this so you understand where we take it from here. We're covering we pretty much are sampling 94% of the seagrasses in Texas. So 94% of the seagrasses in Texas are distributed from basically San Antonio Bay or south of San Antonio Bay, Aransas Bay, to the border to Brownsville. And a good 40% of those, maybe 50%, are in the lower Laguna Madre. So along this, in this area, we have implemented what we refer to as a Tier 2 monitoring program, in which we have taken the best seagrass maps that we have for the state right now, and overlaid on those maps a stratified but random to achieve a stratified but random sampling design. Overlaid this tessellation of hexagons, which are 500 meters on a side. This is, if I know one person in this audience recognizes this right away. Anybody that works or is employed by EPA should recognize this. Barbara Keeler. This is your remap program, and this is, in fact, EPA supported this in the early 2000s. We had a very nice grant from EPA to develop this entire method that we're using now, and it's worked out just beautifully. So we're occupying now over 600 stations along the Texas coast, and we occupy these during peak leaf biomass or peak cover during the early fall. And we occupy, again, those stations and basically from now San Antonio Bay all the way to to Brownsville. We we have broken some of these areas up into small subregions so we can understand better some of the patterns that we're seeing and relate some of the trends to abiotic events. One of the most interesting events that we noticed here was from 2011 to 2013. We saw a dramatic change, unexpected change in Seagrass, Saren Gaudium filiforme cover, and this is manatee grass. So you can see here all the warm colors are high percent cover. So the reds are nearly 100% cover, and when you get down to blues, you're down at 5%. But this is Saren Gaudium cover in Upper Laguna Madre. And you can see the area, it's a mix of blues, yellows, and reds, and grays. And by 2013, nearly all the Saren Gaudium is gone. Just disappeared. And we were shocked to see this. At the same time, we noticed that Haloduli radii, which was, you can see here, all the reds and yellows for Haloduli, came in and its cover increased substantially to make up for all that bare area that was up by Saren Gaudium. This is a fairly big event for Upper Laguna Madre, and we still got some good ideas on what caused it. In fact, we're getting ready to submit a paper next week on this, but we're fairly convinced that it's related to salinity changes. So Saren Gaudium this is where knowing something about the physiological ecology of the plants becomes really important. There are 5 seagrass species in Texas, and of those 5, I would say that Saren Gaudium is probably the most sensitive to salinity. So for a 9-month period in late 2000, I'll bring this next one up, this 2012 to, let me get these salinities, late 2012 to early 2013, we had a period of salinities that were consistently above 50 to 55 parts per thousand and lasted for a good 9 months. Before that point, they were high, but they jumped back and forth between 30 parts per thousand and 50, and truly variable as they usually are. But this period here preceded the loss of Saren Gaudium. And when I arrived here in '88, there was no Saren Gaudium, or '86, there was no Saren Gaudium in Upper Laguna Madre. Didn't exist at all. And that's because the lagoon had been a very hypersaline lagoon for many, many decades. And only recently has that those salinities ameliorated a little bit. And so Saren Gaudium started to invade and then took over. And now, after this period of hypersalinity, it's back down to very low covers again. This is important because, as I'll show you in a few minutes, these seagrass species have different architecture, as you saw, and that means they have very different capacities for carbon storage. So, in terms of blue carbon, we know that sediment and living biomass are really important carbon stores, and seagrasses, although they occupy only 0.2% of the oceans, they probably store store enormous amounts of carbon, about 10% of the total carbon. I think if I'm quoting correctly. And then there's, of course, carbon-bearing sediments plays an important role through that biogeochemical cycling I was just showing in the very first slide. All those crazy arrows and boxes. Those become very important processes to understand. So Texas seagrasses, again, just as a brief overview, what you saw, the ones that, the plant that with the most robust architecture is certainly this one, Phalasia testudinum. Very thick roots, rhizomes, a lot of carbon, a lot of starch stored in this. And I hope, Kim, you did the demonstration with all my students. I always eat my plants. They should have been chewing on it. Oh, they're still good. They're ripe then. So if you take a piece of, Julie will do this, she'll take a piece of rhizome from Phalasia, and you can chew on it, and you can taste the starch. It's got a lot of good nutrients in it, a lot of fiber, right, Jace? It's good for you. But it's very robust, and you can taste that starch. But we look at these, they're much more much finer plants. And there's not they're not as long-lived. Velocity is a plant that can go for 10 years, and these are basically weeds. They come and go in your front, just like in your front lawn. So it's important. So it's important to appreciate that, because Thalassia turns out to store quite a bit of carbon. So what we refer to as our Tier 2 methods. We go out. There's up till this year was 567 stations. Now it's over 600. We go out to all these and we do water quality work. We do seagrass height, percent cover. We put everything in GIS. We do carbon, nitrogen, phosphorus ratios. We do Del-C13, Del-15 values of the tissues. We get all kinds of neat data, which is all, by the way, published on a website, texasseagrass.org. So what we did for the blue carbon, we said, okay, we have all this percent cover data, but we don't have any biomass data. So we need to start building a model to relate biomass to percent cover. So we started doing that this fall, and we're just getting started. These are just rough estimates, but not bad. So we're regressing here, total biomass, pick your plant, or particular species, and we regress below ground biomass against percent cover, above ground biomass percent cover, and here's a graph of percent cover with total biomass. And using these relationships, we can then go back to those nice seagrass cover maps we have all over Texas and start estimating, saying something how much biomass we have. So here's the first attempt at it, and this is literally how the process is. We've been doing this for the last 72 hours. Because it's very, we've been working on it for a while, but we haven't had time to put it together, so nothing like giving a talk, having that pressure to do it. So you can see here, again, the warm colors are higher amounts of carbon storage. These are now we have carbon in grams carbon meter squared. So you can see the warm colors are hot spots, the blues areas of lower carbon. The range here is from 0 to 276 grams meter squared. And you can see some of the patterns. You can see some hot spots, a lot of hot spots here in Upper Laguna Madre, and a few, of course, in Lower Laguna Madre, and some areas of lower carbon as well, and even down here, but in Lower Laguna Madre, although this is a little bit misleading because of the scale. So that's the first order. That's kind of interesting. And now let's look at it by species. Now, to the left is how we do in Saren Gaudium. And we group these because these are these delicate plants that have very similar architecture versus Thalassia. Note here, from blue to red is a 0 to 75 grams per carbon per meter squared range. And you can see the distribution of those blues and reds along in Upper Laguna Madre through the coastal bend. And this one is for Thalassia testudinum. And this one goes from 0 to 276. So we've had to use a much bigger range for Thalassia because it's storing a lot more carbon. Now if we move on, and this graph, this table, is an example of the kinds of above ground and below ground biomass we see in terms of, now we're in megagrams of carbon per hectare. So we've scaled up to hectares, and we're about to scale up in the next graph to the entire Texas coast. But this is what you'll find very interesting. Let's pick Thalassia testudinum of Corpus Christi Bay, CB, 5.2 above ground. Below ground, 13. 13 plus 5 is 18. 13 divided by 18 is about 2/3, right? So 66% of the biomass in Thalassia is below ground in Corpus Christi Bay. When you look at a grass bed, you're only seeing 1/3 of the total. Let's look at this one. 11.1 and 28. 28 plus 11 is about 40. 11 divided by 40, 25%. 75% of the biomass of Thalassia in Lower Laguna Madre is below ground. You're seeing, when you look at a grass bed, you're seeing 25% of what's really there. So that gives you an example of just how important this below ground biomass is. And as you go further tropical, it goes even higher. And the reason for that is because of nutrients. When I take my class down to the Yucatan, the plants, the above ground biomass is even, the plants are short, they're stunted, but but the roots, you can't even push a knife into the sediments. There's so many roots and rhizomes. They're photosynthesizing like mad, storing carbon, but they don't have any nitrogen to make chlorophyll. So that there's not a whole lot of above ground biomass because there's hardly any nitrogen to build pigment proteins, which is the above ground part. For Haliduli radii and Saren Gaudium, we can look at Lower Laguna Madre here, 5.2 and 9, that's about 14. Again, 5 divided by 14, about 1/3. So again, 66% of the biomass is below ground, even for these weedier species. So in general, there's quite a bit of biomass below ground. And the reason why that's important is because various folks have shown, including a couple scientists here a few years ago, that roots and rhizomes and seagrasses release a lot of dissolved organic carbon. And that dissolved organic carbon ends up in the sediments, it ends up in the soils, and becomes locked into soils and therefore the sediments and becomes a very very important carbon reserve as well in the sediments. Okay, so let's move on from here and let's look at the entire coastline. So now we've taken in the first column here the hectares, the total number of hectares in Texas and in these different regions. This is the coastal bend, Upper Laguna Madre, Lower Laguna Madre. You can see how many hectares of seagrasses in Lower Laguna Madre. This 83,000 hectares represent 830 square kilometers. That's a lot of seagrasses. That's a lot of seagrasses in Texas. Now, what we did here is you want to know, well, how we've been doing this for 5 years now. We just finished the 2015 analyses are just finishing. So we want to know, was there any differences between over the last 5 years, from 2011 to 15, just for fun? And sure enough, when we look at the big picture here, we actually do see a difference, and it went up. So we actually have a higher amount of storage in 2015 in total biomass, above and below, than we had in 2011. Whether it's significant, I doubt it. It's probably in the noise, but at least it didn't substantially decrease. When we look at the whole Texas coast, we can see areas of yellow and reds, and then we see this blue area down here in southern Laguna Madre that is a little bit lower. But first, you know what, I have to go back one slide. Let me see. No, that's the wrong way. Here we are. The reason I have to go back one slide is I need to point out to you, in terms of the amount of carbon that's stored in Thalassia on a hectare basis versus Saren Gaudium and Haliduli. Assume that this is 100% cover. Look at the difference. 13. This is on a standard aerial basis, so we're comparing apples to apples. 13 versus 5, 28 versus 9. So Thalassia stores a lot of carbon, So ideally, of course, when we see losses of Thalassia relative to the other two species, we're basically losing carbon, really speaking. So back to this one. This blue area here is kind of interesting. This is a decline. This is a net loss. So this graph represents differences 2011-15. The blue represents a loss versus the yellows and reds. And why is that? Well, why is that is right here in this picture right here. So so Thalassia testudinum dominates the Lower Laguna Madre, particularly the very southern extent. You can see the high percent cover of Thalassia in 2011, and you can see that's declined somewhat through 2013, and 2015 looks about the same. So we did lose Thalassia cover, and that translates to a larger, a very discernible decrease in carbon storage because Thalassia stores so much. So those are the things we need to watch out for. Those are the things that we need to be aware of. In summary, we have a very high, I need to go back one more time because I want to go back to that 300 teragrams. Oh, darn. So look at this number here. So the total teragrams of carbon stored on the Texas coast in terms of seagrass biomass is about 2. Remember earlier I talked about, I gave you the number 300, loss of 300 teragrams of carbon for seagrasses, and so this is only 2. So you can imagine losing the area the size of Texas in seagrasses multiplied times 150 times in terms of what we're seeing worldwide. It's a pretty serious problem. So that too represents 800 square kilometers of seagrasses. 800 square kilometers. And that earlier slide I showed at the very beginning, we talked about loss of 300 teragrams of carbon. So that puts it in perspective a little bit for you. Okay, so summary, we have, of course, potential for very high carbon storage. We've got gains and losses, but pretty much steady state at this moment. Our next step that we're very excited about, we've wanted to do, is we're going to start doing the sediments, the soils, at all 600 stations. So we're going to have a very good estimate now of soil carbon storage by this time next year. So again, I mentioned some of the high points. Below-ground storage is a critical carbon reserve, not only for us, but also for the plants. That's how they keep going. And there's important differences in species composition with respect to carbon storage. With that, these are the folks that have funded this program over the last 5 years. Mission-Aransas NER, the NOAA program, the Coastal Bend and Estuaries program, the National Park Service, the General Land Office, and also in-kind support from the Nature Conservancy. So with that, it's a great time to have questions. Thank you.
Thank you so much, Ken. Any questions, guys?
The losses in turtlegrass and the lower salinity related?
Oh, Bo, yeah, so the question was, is the loss of seagrass and lower lagoon madre salinity related? No, it looks like we're looking at the CNP ratios of the plants. It looks like we're seeing some evidence for eutrophication or nitrogen enrichment that might be causing epiphyte blooms, Bo, or some local algal blooms that are having effect on the plants. This epiphyte coverage is one of our biggest concerns, and there is a sewage treatment plant in that area that we have that we know about. So we're not really sure, but we kind of suspect something dealing with nutrient enrichment. Yeah, good question. Yes.
It's really great to see quantification like this and get appreciation of seagrass down here. So trying to make sure I keep the units in my head right. So 2 teragrams is 2 million tons. 8 million tons roughly of CO2. I'm really curious. So that's the living biomass. I know you didn't work in soils, but
Right.
from your experience in the soil, what's your sense of the transfer of living biomass into soil organic?
Well, that's a good question. I wonder what it is on those linkages might be, on an annual basis. But, soils are generally, sediments, marine sediments are generally 2 to 3, and these sediments are probably at least 2 to 3% carbon. So we can start doing the math in our head in terms of how many 800 square kilometers times and do some estimates on the weight of sediments in a square meter. And also there's issues of how deep, we are going to figure out, we are going to look into the vertical profiles, the vertical distribution of organic carbon. That's going to be very interesting too. We have some nice deep cores that we plan on taking, but I would say, just based on what we see in the literature, that the carbon storage in the sediments is going to be darn close to equivalent to what's in the plants. What do you think? You're much more an expert on this than I am. You've been in literature where I've not been in the literature too deeply on this yet.
Yeah, I'm just really curious because there's a lot of regional variability, particularly in seagrasses. Oh, thank you. There's a lot of regional variability between seagrasses, and my gut sense is that if you're in a lagoonal system like this, there's probably, the sediments are probably a bit finer. There's probably an opportunity for better carbon storage.
Yeah.
Particularly with the species which are putting down those rhizomes.
I'd say so, yeah. And these islands, it turns out, yeah, you made a great point there. These protected lagoons. So if you look at some of the geological background for this area, the island that we're on now, Mustang Island, this island has been here for at least 7,000 years, whereas the islands on either side of us, Padre and even St. Joe or San Jose, have been only around for 3,000 or 4,000 years. So that means we have been protected for the last 10,000 years, that this lagoon has been relatively protected, and there's a huge amount of carbon that's been just stored and dumped and been sitting there for a very, very long time. So I agree with you. I think they're going to be, these types of systems are going to probably store a lot more than an open Barrier Island system, where there's a lot of more open Barrier Island system, which has a lot of turbulence and ocean energy, wave energy going through it. Yeah.
So just to make the last point, so
Yeah.
from what you just said there, then even though the numbers are great for the living biomass, it might just be the tip of the iceberg, as it were, or the mudberg.
Yeah, no. Mudberg. No, yeah, I'm very interested in seeing what we come up with for sediment, organic carbon in these systems. We know it's high, but to actually extrapolate over the entire coast, it's going to be really interesting. But I think they're going to dwarf the seagrass carbon itself. Yeah.
I promised I'd ask you this question. Because we were talking about it outside.
Yeah.
When we were looking at the seagrasses, and it may be a dumb question, but when the photosynthesis that occurs, most of the seagrass is below water,
Yeah.
right? So where is it absorbing the CO2 from the water, or can you talk more about the chemistry of the photosynthesis in seagrasses as compared to terrestrial plants?
I'm a marine botanist. We could be in trouble because I could get off. This is my favorite topic, and we could be here for a couple hours. No, a few years ago, the difference where C3 versus C4 plants. I'll start the story there. C3 plants, like grasses, sugars, they fix carbon and following the typical Calvin cycle, everything's great. C4 plants are exemplified by corn, and corn has and cactus and cam plants, C4 cam plants, have these adaptations to reduce water loss through closing of stomata, and they have an additional carboxylation pathway to grab CO2 without grabbing oxygen to reduce water loss. Well, C3 and C4 plants, because of that, have different carbon isotopic signatures. C4 plants have a lot of C13 in them because they don't discriminate against the lighter isotope, whereas C3 plants, like grasses, have a lot of C12 carbon in them. And in fact, my professor here, what was interesting is that years ago, my advisor, Pat Parker here, made a lot of money by helping USDA sue folks that were claiming they were making maple syrup when in fact it was corn syrup laced with lots of food coloring, and all he had to do is measure the stable carbon isotope ratio of the material. The reason why I say this is that for a really long time people thought seagrasses were C4 plants because they have a lot of C13 in them, like corn, and they were convinced that they were C4 plants, had this additional carboxylation pathway, which they took up CO2, aqueous CO2, and fixed aqueous CO2 in photosynthesis. Turns out that's completely wrong. They don't have there's no evidence for this additional carboxylation pathway, and what they do is they take up bicarbonate, HCO3. And it takes an extra energy, but they take bicarbonate across the epithelial cells, and they dissociate it using ATP energy, a couple ATPs, into CO2 plus a hydronium ion, and then that CO2 is then released inside the bundle sheath cells or in the epithelial cells internally and enters the Calvin cycle. So their carbon source is not aqueous CO2. It's bicarbonate, and bicarbonate has a lot of C13 in it, so that's why they have a high C13 signal. So that's how they photosynthesize. That's their carbon source. It's very expensive, but they've got enormous amounts of light, and they're photosynthetically very, very productive. I mean, they're among the most photosynthetically productive plants, land plants, or I should say flowering plants around. So that's the short answer. I'm not sure how much more you would like to know. about photosynthesis. I mean, that's basically where they get the carbon from, and they store a lot of it in terms of the carbon reserves in the below-ground tissues, and then the rest of it they use for above-ground tissue with taking nitrogen out of the water. They get 50% of the nitrogen from the water and 50% from the sediments, and that's how they make pigment proteins for the above-ground biomass. And they don't like cold temperatures. They really die off in the wintertime, whereas they stay very active at lower latitudes, like in the Caribbean. There's biomass year-round. So, did that help? Did that answer it? Okay. I know
right,
the
any
graduate
other questions?
students. Come on, Ray. Oh, Victoria, you must have a question. You wrote this talk. All right, well, I think it's time for your break anyhow, right, Dana?
Yes,
Or some kind of break.
We are going to do a break, and it's going to be a fun one. Because A, it's a break, and B, we're going to get you to...